Welcome to ecotopiaumedia. This is Melissa McGinnis, your host. I'm joined by my co-host, Tom Wright, and we have a lovely, lovely, lovely friend joining us today via Skype, John Fagan. Nice to see you again.
Oh, great to see you. It's great to be here.
Dr. John Fagan.
Oh, excuse me. I had no idea. Well, I mean, I should have known.
Yes, you should have known.
Well, thank you so much again for joining us. I know that you're doing some work with GMOs, as it relates to potentially being a proponent for non-GMOs. But tell us, when did you first learn about genetic engineering?
Well, that happened in I think it was the last year of my graduate work at Cornell University. And this guy, Paul Berg, professor from Stanford, came each week. We would have a seminar on Friday afternoon, and we'd have a visiting speaker who would come in. Paul Berg came in, and he talked about the very first experiments that had been done with he had done with recombinant DNA research.
Explain a little bit to the audience what that means.
Yeah, I will. What
thank
he
you.
showed is that he could take a piece of DNA, a gene from a virus, and insert it into the DNA of a bacterium, and therefore create a bacterium that could actually, at least in principle, could express that DNA, express that gene, that viral gene in the bacterium. And he showed all these electron micrographs and all these things proving that he had been able to cut and splice DNA this way. Now, he wasn't really showing that it could express effectively, but he stood on the stage and explained that this was the beginning, and that there was, that where this was going was taking us in the direction of being able to cut and splice DNA from different organisms, put them together, and express new functions in a bacterium that have never existed in that bacterium before, and potentially new functions in human beings or in any other organization or organism. And it was quite an eye-opener for me, because the way that biologists and biochemists have been studying living things up until that point in time has been like a computer that has read-only memory, and that's it. In other words, it can do what's already been programmed into it, but you can't put any new information into it. And a computer can be useful for something like that, but it's only when you can reprogram it that it becomes something that has the power and flexibility of information technology that we have today. What happened with recombinant DNA technology is that we learned how to reprogram the living organisms, how to put new information in and have that expressed. And to me, at that point in time, especially in my PhD work, I had been looking at how cancer happens, what causes cancer. And we were able to use all sorts of cell biological mechanisms to sort of diagnose what was going on. But it was always, you'd come down to the end of the experiment and you'd say, well, it could be this, or it could be this, or this, or this, or this, and probably this. And so we'll do this next experiment. So it was always not definitive. And what I saw was that with recombinant DNA techniques, you could become definitive on the level of the genetic program of the organism itself. And that was powerful. And it was like it just opened things up for me. And it was, when I finish here, I'm going to go do that. I'm going to go do genetic engineering, because this is the future for biology. And in fact, personally, I still feel that genetic engineering is a very powerful tool for understanding biological systems. So there's no doubt about that in my mind, or any ambiguity about it. It's a powerful and very useful tool, because it allows us to understand things about living systems that we could never learn before. So after I finished, I went on to the National Institutes of Health and began to actually clone genes and move things around and do these sorts of things and begin to understand new things about biological systems, particularly as they related to chemistry. But always there was this nagging thought, this nagging little thing about, well, this is a technology that's really powerful. And am I using it responsibly? And is it something that, is it being used by others responsibly? And what is its impact going to be on our world? So always that was there. And then about the 4th year I was at NIH, I was invited to Korea. And I was asked to give 2 talks. One about my research using genetic engineering to study the cancer process, study carcinogenesis. The other was to talk about about biosafety, how these things could go wrong. And I still remember the first talk, the room was packed. The second one, I would say at most 50% of the people were in the room. And afterwards, it was so funny because afterwards I just commented to my host, I said, the room was half empty for this talk. I'm wondering, where is everybody? And he says, oh, John, they love this technology. They want the technology, but they're not really interested in whether these other potential risky impacts. They just want to do it, and that's really how this thing has evolved. And that's where really I began to have concerns. And over the next few years, the same kinds of things continued to come up. And at one point, there began to be a lot of sort of promotion of using genetic engineering for modifying the human genome. And these scientists were talking about how this could be used to cure diseases. But I knew that in fact there were simpler ways to diagnose whether, instead of re-engineering an embryo, well, you take 10 embryos and you diagnose and find out which one is damaged and which ones are okay, and you reimplant the okay ones and let the others go. That's the kind of thing that's much safer to do. So I knew that there was hype here, and it became clear over time that they were much more interested in genetic improvement than gene therapy. They were seeing a big market for the Mozart gene, and for the Wilt Chamberlain gene, the genes that are going to make your child the best athlete in the school, and your child the most talented, musician, or the Einstein gene, which is going to allow them to do this and that in science, So I became very concerned about this and did some really deep thinking about it, and talked to some of the people that I consider my mentors, and came to the conclusion that it wasn't a good thing to allow this to go forward. And so I took a public stand and actually gave 2 grants back to the NIH. One, simply I'd already received it and I returned it. The other one, after I'd returned that one, the other one was in process, and they were thinking to give it to me. They called me up and said, well, now, we hear you gave this grant back, So here they are, we hear you gave this grant back. If we gave you this other grant, would you do the same thing? And I said, yeah, I think I would, And so, never heard from them again. And so, basically, I took a public position. I did a press conference in Washington, and all 3 networks. Then there were 3 networks, not 4.
What was the year, approximately the year?
It was 1994.
Okay, 94. Okay.
And if you look in Time magazine, early in that year, you'll find some articles like one of these guys saying, just give us another year or two, and we'll be able to, if you have a problem, we'll give you a snippet of DNA, and it'll solve it for good.
Oh my goodness.
That was the kind of thing they were saying that was so blown out of proportion, creating expectations for people that are so unreal, and at the same time creating a market, which was to me the big concern.
Yeah, market based on wishes and dreams.
Yeah, it was wishful thinking. A lot
Yeah.
of the same thing that we see with genetically engineered crops today. You know, they've been talking about how they're going to be resistant to drought and to do this and that, climate-ready plants. But the reality is 98% of the crops out there today are either herbicide tolerant or they produce pesticides, the BT.
Yes.
And the reason that there's so much of that and so little of the other is that they failed to use genetic engineering effectively to do these other things. And there's a reason for it. It's that genetic engineering techniques, even the most recent ones like CRISPR, you're hearing about how wonderful CRISPR is. These things are not precise enough to safely change these crops in a predictable way. And as a result, you can do some little things like create a plant that is resistant to an herbicide, but to actually create a complex trait like like drought resistance or resistance to a certain disease or things like that, it's really hard to do, and they failed. They've been saying they're going to do it for 20 years, and they failed.
But they keep saying it because of the money keeps flowing.
Right. It's just a hype. It's just to sell it.
Yeah, that's fascinating that way. So explain to the audience these two main things that are on the market: the herbicide resistance and the BT. Explain what they are and which crops are using the technology.
Okay. So the most famous one are the Roundup Ready crops. And these are crops that have been genetically engineered. They put a gene into these plants that originally came from bacteria, and it makes them resistant to an herbicide called Roundup. The main chemical in it is called glyphosate. And this herbicide kills all plant life, essentially. And when you engineer the plant so that it's resistant to it, it makes a very convenient model for agriculture. What you do is you plant the crop, you let it come up, the weeds come up with it, you spray the field, you nuke the field with this stuff, and it kills the weeds, but the crop doesn't die. Now, the crop is not impervious to it, and in fact, any farmer will tell you that for at least a couple of weeks after you spray, the plant is not healthy, and it takes some time to recover. And in fact, it has an irreversible effect on the plants over time. But it's enough so that the differential is enough so that you can very conveniently kill the crop or kill the weeds and protect the crop in this way. So they call it Roundup resistant crops, and it's very it's used in many countries around the world now.
Now there's some feedback loops that evidently are occurring. One is it not true that certain weeds, they call them now super weeds, as a euphemism, they become resistant to the Roundup? They actually genetically evolve themselves.
Right. And so Darwinian selection steps in here, and over a few generations, the weeds become truly resistant to the Roundup so that it will no longer kill them. And so in many places around the world, these Roundup Ready or these herbicide-resistant crops are failing because of weeds. Here in the U.S., there are something like 27 different, I guess around the world, there are 27 different weeds that are very noxious weeds that are resistant. And there are some places in Georgia where they've used Roundup Ready cotton for many years, and the weeds, the resistant weeds, have gotten so bad that they've had to essentially abandon those fields. Some of the weeds are like 6 feet tall.
Oh my goodness! No wonder they're called superweeds.
Yeah, they really are super weeds.
Little jungles. So it's soy, corn, canola, cotton, and sugar beets that they're using this.
Sugar beets, and also, yeah, those are the main ones that have been engineered with Roundup resistance.
And explain to the audience that the seed from the genetically engineered crop is useless. Explain that to the audience.
Well, yeah, it depends. If it's hybrid seed, then it's useless. But in fact, in fact, they use that as a technique for protecting their intellectual property. But with some crops, the engineered seeds are quite viable. For instance, like in soy. So that happens. Now,
so are farmers allowed to use the seed from the crop?
No, if they do that, they get sued. And Monsanto has in fact sued hundreds of farmers over the last decade. Hundreds of farmers who had naively tried to regrow either their Roundup Ready soybeans or Roundup Ready canola. In fact, there's a famous case in Canada where a farmer, Percy Smeissner, had done this, and he, it ended up sort of as a draw. He fought to the death about the whole thing, but it was, it's a very difficult situation.
And he was a famous canola seed breeder, and he wasn't growing GMO canola.
Right. It came in, it drifted into his fields, and it essentially polluted it. And so he was sued, not because he was actually intentionally growing it, but because it had polluted his field. And this created a precedent that is quite concerning. And it's, if you're a farmer and there's a risk of that kind of cross-pollination, it creates big questions in your mind, especially when you have, when there are Monsanto agents out there, and there are Monsanto agents out there looking for farmers that are, violating those patents. So it's a very tricky situation.
John, when you mentioned that these GMO promises failed, what were the expectations? I mean, was it higher yields, healthier plants, and we're seeing that actually the plants are inferior after they get sprayed for a couple of weeks, and if the weeds are becoming resistant to the spray, right, then where are the promises and where are we at today?
Well, they had promised higher yields, more nutritious crops that would be resistant to pests and diseases, crops that would be resistant to all sorts of stress, whether it's low nutrients or not enough water or too much water, flooding, that sort of thing. So they've promised all of these traits, but in fact each of those traits is quite complex. It doesn't involve just one gene. So they're capable of sort of putting one new gene or a couple new genes in a crop to give it certain agronomic traits like resistance to herbicide or producing their own pesticide. But when you have these complex sort of highly sensitive regulatory systems that would need to be engineered, they fail to do that. The technology is not up to the job, and therefore they've simply not been able to achieve what they wanted to. So,
and I'm sorry, go ahead.
Let me say the last one thing. Today, even though you hear over and over again in the news about how genetic engineering is expected to improve the yields, there is not a single GMO out there that consistently delivers yields that are better than the corresponding non-genetically engineered crop. It simply has not been done.
Hasn't happened. Not to mention what happens to the soil of being sprayed with so many
The
herbicides.
soil chelates, doesn't it?
Yeah, it pulls, from what we understand, glyphosate is a chelator of important minerals that are important for the plant and also for animals or human beings that eat that plant. So it's reduced in its nutritional value because of the Roundup in that respect. And also, the most concerning thing is that Roundup now has been patented as an antibiotic. Actually, Monsanto has a patent on Roundup as an antibiotic.
Sorry for laughing, but go ahead.
I know. And so it kills the microbes in the soil, and it also disrupts the microbes in the digestive tract of livestock that eat it. There are studies out there showing that this happens. So, it's not good for you, and it's not good for the soil. Soil is a living thing, as both of you are well aware. And if we're killing it this way with Roundup and also with other pesticides and chemicals, it's not going to support the quality of food that is going to really be good for people, for our world that way.
So it's a fail, fail, fail.
Yep.
Triple fail.
Triple fail.
All right. Well, thank you for your time today. I know we went a little bit longer, but I think that we really learned a lot about GMO that there was no clarity on it. And I think you really, really did a great job for ourselves and for the viewers. Thank you, doctor.
Oh, it's a pleasure.
Okay.
Thank you.
Thank you.